I'm Anne-Marie Fauvel, as Net just mentioned. I'm the tech team coordinator for the Be Informed Partnership. And I have to tell you, I have the best job in the world. I get to work with the tech transfer team, which are field specialists that experience bees like no others. And it's really exciting to learn from them. I also get to collaborate with some of the most dedicated and successful honeybee researchers in the US and even in the world. So that's even more exciting. So you will notice that specifically some of those researchers will be very involved in the presentation today. And specifically, you will notice that I have some slides in my webinar portion anyway that are courtesy of Dr. Marla Spivak. And I must thank her for the wealth of information that her research has generated in this field. So without further ado, I thought I would get started with just a quick review of what our organization does, just in case you have missed some of the webinars that we've had previously. So the Be Informed Partnership is a small but impactful nonprofit organization, and we work out of the University of Maryland, although, as Net mentioned, we are now pretty much spread out across the US, and not just the US, but we even have people in Canada and in Mexico. So we're really kind of very spread out. Our reach is fairly broad, and we work with a big extended family. We work closely with about 10 universities, as you can see some of those universities listed here on the slide. And we have about 12 to 15 full-time staff members at any given time. And so that gives you an idea of how small organization, but how broad our reach is. So our main mission is to improve colony health. And we do this by supporting beekeepers and by bridging the gap between science and industry. And in doing so, we found ourselves to become the largest US repository for data, colony health data in the US. So we run multiple programs, including the annual National Honeybee Loss and Management Survey, as you may have been on one of those webinars where we gave some of those results earlier, about 6 weeks ago. And then we also have the Sentinel Apiary program, which we also presented on, and the Tech Transfer Team program, working mostly with commercial beekeepers, which we pool from their observation and their data recording quite a bit. So that's what we're going to do today. I must say that if you find it important or see value in the work that we do or the information that we disseminate, please visit our website and consider a donation to the Be Informed Partnership at beinformed.org. Okay, so with that, let's start with a poll, because I want to know how many of you would be willing to purchase hygienic stock. So how likely are you to purchase hygienic stock of honeybees? Very likely, likely, neutral, not likely? Let's see.
We'll give it another 15 seconds. All right, we're at about 80% participating. I'll share the results.
Thank you. So this shows me that about 50% of the viewers here are very likely to purchase hygienic stock, and the other 50% are maybe a little less likely, or maybe not as excited about it, or maybe don't know much about it. So that gives me a good idea of how to kind of continue on. So let's see if we can convince you to all become very likely throughout this presentation. So here is what we have in mind today. We will start by discussing the basic concepts of honeybee health care and defining hygienic behavior. And once this is established, we hope to justify the need to improve this hygienic behavior in honeybees. And we will review the ongoing breeding efforts as well as programs, and also some of the literature to better understand the mechanisms involved in hygienic behavior. And then I will pass the baton to Rob Snyder, our California field specialist, who has been performing hygienic behavior testing with some of the California queen breeders for 9 years now. And so he will describe to you how the test is actually performed, how the results are read and calculated, and show us an overview of the data of the past decade and discuss the trends in hygienic behavior in California queen breeders. We will also discuss a few questions that we would like to kind of pursue and look into our database to find answers to. So without further ado, let's get started. I made this slide before the pandemic, so that's when we used to see signs mostly instructing employees to wash their hands before returning to work. But of course, since then, signs for everyone's to wash their hands are now everywhere. And so this kind of leads me to ask the question, what does it mean for a honeybee to be hygienic? So poll number 2, real quick. Let's see. I want to know from your quick view of the previous slide, what is the bee doing in the photo? The bee is washing her hand, the bee is grooming herself, the bee is performing the waggle dance, or what bee? All right, we're going upwards. Place your votes.
Give it about 5 more seconds here. All right, I'm going to end the poll and share the results.
All right, so the majority of you, 74%, the ones who voted anyway, said that the bee is grooming herself. Okay, so I'm sorry, this was a trick question. And so I love the image so much that I found myself fooled by it. And grooming on this photo that you see on the right-hand side is not a bee, but it's a fly. So it was a trick question. The answer was really what bee? And it seems like 20% of you figured that out, which is great. If you find yourself having difficulty recognizing sometimes bees and flies, I've just put a picture on the left-hand side to kind of show some of the differences. The mimicry is really good, isn't it? So one of the ways to recognize them easier is you look at the antennae. The antennae of the fly are much shorter than the antennae of the honeybee, which is on the left. And then the wings are also very different. So the wings on the fly, it's only one set of wings, and they're usually longer than their body. And on the other side, you have the honeybee with double set of wings, which is usually shorter than their body. But that's a little bit beside the point. That was a tricky question, kind of trying to get you all to pay attention. So let's get on to hygienic testing. Just like washing your hands is a preventative measure to avoid contracting certain diseases, Wash Your Hands campaign, just like the vaccination campaigns, are all effort that are part of our own collective defense against pathogens. And the term social immunity is defined by the act of mounting a defense against pathogens that benefit the individual other than the main actor. So in other words, the efforts that are targeted toward the whole of society or a different individual than the one who is performing them. So the baboon on the left-hand side here is pulling parasites off another member of the community and is doing so for the benefit of the other individual and probably the benefit of the entire community by lowering the parasite load to begin with. Leaf cutter ants also farm their own fungus that they eat, and they mount an antibiotic treatment to keep the fungus garden free of infections. So those are all examples of social immunity. And this type of immunity can be observed in most social insects community, like ants, of course, termites, and our beloved honeybees. So I want to take 1 second to talk about pathogens and the way they work, and then the strategies that social immunity actually come in to counteract those pathogens. So for a pathogen to be successful, it needs to achieve 3 things. So the parasite needs to move from the outside to the inside of the nest. It has to enter, right? It has to get in and go past the barriers, the first barriers of defense. Once it has entered, then of course it has to survive and it has to establish itself. So survive and stay healthy, thrive. And then more than this, then the pathogen needs to reproduce, multiply, and spread, of course, to the rest of the colony. And this last phase is when the pathogen takes up the potential to become infectious. That's when an infection starts, when you have lots and lots of agents out there to cause damage to the colony. So on the other hand, to neutralize the success strategies of the pathogen, social immunity success strategies work to counteract the pathogen strategies at all the levels. So of course, the first level of defense is to avoid the pathogen to begin with, to prevent it from accessing the nest. That's the first step and the first line of defense. And that's the base case scenario. Of course, if that doesn't work, then the next step would be for the social immunity strategies to reduce the number of agents or pathogens in the colony and prevent an infectious stage. And of course, when that fails, after that, if that fails, then you have a lot of pathogens. The ultimate goal for the strategy for the community, for the colony, is to eliminate that pathogen altogether. So I have one more poll for you in my portion, if you want to take that poll. I think that's poll number 4, and I think that we kind of messed up the number, but that's okay. How do honeybees avoid pathogens? So here are a couple of different things for you to choose from: grooming, sting and venom, collecting propolis, producing antimicrobial secretions, all of the above, or none of the above.
Another 10 seconds.
I want some votes. Come on, let's get to it. At least 80%. We're at 70. We're almost there. Just a couple more votes. All right. So it looks like a good majority, 70% of you, mentioned that all of the above, so grooming, sting venom, collecting propolis, and producing antimicrobial secretions, were all ways that honeybees avoid pathogens. So I'm going to address this in the next few slides, but let's just say that a majority of you have a good consensus on some of those things, and I think that you're on the right track. So Dr. Marla Spivak uses Sylvia Kremer's work on ant colonies and expands it to honeybees. Kremer has detailed social immunity in ants and coined the term care-kill strategy of social immunity. I am aware that the term kill may make some of you cringe a little bit, but I will use it as it is originally intended to really describe insect behavior. So let's keep that in mind. So the care-kill strategies are a set of behavior that collectively make up the mounted social immunity of a colony of insects. In this case, in ants, care strategies, which are meant to prevent an infection from happening, care strategies include grooming, antimicrobial secretion, and nest sanitation. And the kill strategy, that's once the invader is in and the infection has taken place, after prevention has probably failed or was not strong enough. Then the kill strategy is to sacrifice infectious individual for the good of the colony and hopefully eliminate the pathogen. So in the honeybee world, Dr. Marla Spivak from the University of Minnesota, along with many others in the US and all around the world, have broadened the care-kill strategy to really specify it for honeybee social immunity. So in the slides to come, we will use multiple scientific publications to build a foundation of knowledge of hygienic behavior. But these two specific publications at the bottom of the slide that you see here are fascinating, and a lot of the information in the next few slides come from those two publications. So what do the care-kill strategies look like for honeybees? Well, here is a simple list of behavior honeybees perform every day to care for their colony and prevent infection of pathogens. And so this is to answer the poll earlier. So grooming is one of them, right? Bees groom themselves and each other. Of course, they go from flower to flower. They touch a lot of doorknobs or surfaces, if you will. And then grooming is just like hand washing. It's one mechanism to prevent infection, first line of defense. And then they also guard their entrance and the whole nest, and they will use venom to neutralize some of the bigger pathogens and enemies, right? And then they also use antimicrobial secretions that they incorporate in their food and in royal jelly. And they possess gut microbiota that confers that confers some immunity to pathogens in the gut and also in their pollen in the bee bread that they make. And then, of course, finally, honeybees collect resins from trees, propolis, as you all know, that's the sticky stuff. And they use it to coat many of the surfaces in the colony, nooks and crannies in the colonies, to prevent the pathogens from from entering the nest. But it also looks like propolis possesses many antimicrobial properties. And we're starting to believe that it is extremely important component of the healthcare system of honeybees. And of course, Dr. Spivak has done some work on propolis. I think that there's a lot more to be discovered when it comes to propolis, but there's some really cool advantage to breeding for more propolis collection. And of course, that's the care part, and so that's the prevention aspects. This is how to prevent pathogens from coming in. But if the pathogens do come in, then we have to go to the kill strategy. And that is what we coined the term hygienic behavior in the honeybee world. What we call hygienic behavior is the kill strategy, which involves detecting diseased pupae and larvae or mite-infested pupae and larvae. And then after you detect it, they need to uncap that pupae or larvae, and then they need to remove that or discard that pupae or larvae. And sometimes, instead of doing that, they recap it. And we don't really know why they do this. So we're going to focus on the first three dots, but know that recapping is sometimes happens. So here you can see hygienic behavior in action. On the left image, you see a bee that's actually removing a pupae from the cell. And in the cells just right of that one that she's working on, as well as on the cells on the lower image on the right-hand side, you can see some cells at the bottom where it looks like chewed down. It's like the head has been removed. And so that's what we call chewed down brood. And when you look into your colony, this is something that you might see. And so often in the fall, when mite damage is more apparent, I know that when I open my colony, I see a spot. If I see a spotty brood pattern, then I will look more closely to see some signs of this. If I see signs of hygienic behavior, then that's something that, like on the photo here, I usually pick this up and I see this as a sign of something else wrong that's going on. And so I usually investigate further. So this is kind of something that you could see in your own colony. So hygienic behavior concept is definitely not new, right? Back in 1942, Woodrow and Holst stated that resistance to American foul brood and honeybee colony consists in its ability to detect and remove disease brood before the causative organism reaches the infectious spore stage in the diseased larvae. So we have known about this strategy about hygienic behavior for a long time, but what we didn't really know or didn't understand at the time were the mechanisms behind that behavior, that underlie hygienic behavior. Before I go into the mechanism, I do want to make a little distinction. And in fact, we've had some questions about this to begin with. I was confused about that when I first started, and I hear a lot of beekeepers talking, and interchanging different words like Minnesota hygienic and Varroa sensitive hygienics, and what does it all mean? And so Minnesota hygienic honeybees were bred at the University of Minnesota. That kind of seems relevant. And the breeding program used the freeze-killed brood assay to select which colony to breed from. That's the important part. And the freeze-killed brood assay is what Rob is going to talk to us in the second part of the webinar and how to perform that test. So it is important to know that the Minnesota hygienic line was developed from the result of this test. So the highest hygienic scores were chosen to breed and continue the line. On the other hand, Varroa sensitive hygienic line was developed by Harbaugh and Hubengartner back in the late 90s, and colonies were selected from low Varroa colonies. So the lowest Varroa load colonies were used to breed the next generation of VSH bees. And so in the end, I think that what's really, really important to remember here is that in the end, even though the selection method of each of these two lines were different, both of these lines were selected for their hygienic prowess, so their hygienic behavior. I do want to read you a little quote that I think is really, summarizing of the message I'm trying to put out here this morning. So this is from an installment in the American Bee Journal back in 2008. And Dr. Spivak and Gary Reuter have written this article called "The New Direction for the Minnesota Hygienic Line of Bees, A Brief History." And I read a quote from Marla. "Gary Reuter and I at the University of Minnesota have been breeding honeybees for resistance to diseases and Varroa destructor since 1994, with the goal of reducing or eliminating the use of antibiotics and pesticides by beekeepers. Through our ongoing collaboration with Minnesota and North Dakota beekeepers, we have demonstrated that bees bred for hygienic behavior rarely, if ever, show clinical symptoms of American foul brood or chalk brood and have significantly fewer Varroa mites compared to unselected colonies. So hygienic bees resist these diseases and mites by detecting diseased or mite-infested brood and quickly removing the infected brood from the nest. The removal of the pathogen before it is infectious at the infectious limits disease transmission, and removal of mite-infested pupae reduces the reproductive success of the mite." So I quote, that was Marla Spivak. So this leads us into the discussion of the mechanism underlying hygienic behavior. How do bees detect diseased pupae or larvae? And research has showed that olfactory cues, so odors, are what triggers the series of behaviors associated with hygiene. And so diseased brood would give off olfactory cues, which are then taken up by the adult nurse bees, who will uncap and remove the diseased brood. Hygienic bees are the ones that can detect these cues at very low stimulus. That is, again, the important part here. So what do I mean by that? I mean that all bees possess hygienic behavior, right? But they're not all equal in detecting it earlier on. So in other words, bees that we would consider non-hygienic detect olfactory cues from brood, but only when the stimulus level is really high, when there's a lot of pathogen, when the infection is really high and there's lots of odors, if you want. So that's high stimulus level, it's at the infectious level, and that confers the advantage to the pathogen. But hygienic bees, on the other hand, are able to detect and discriminate these olfactory cues at low stimulus levels. So before it becomes infectious, when there's not that many pathogens. And so it eliminates the threats before the colony is doomed, and that gives the advantage to the colony. So hopefully we're making a good case for hygienic behavior and increasing those behavior in our genetic stock. So Dr. Spivak looked into this even further. This is the latest research that I will be citing today. They wanted to know if the genetic advantage was driven by the enhanceability of the adult nurse bees to pick up on the odor cue, and or the enhanceability of the disease brood to send better and bigger cues to be picked up by the adults. So they designed this really, really cool and fascinating experiment where they put brood of different origins, so either unselected colonies, Minnesota hygienic colonies, and VSH colonies, and they placed them in colonies of the same categories. So for example, in an unselected category, in an unselected colony, you could have brood that came from unselected, VSH, or Minnesota hygienic. And so what this did is that if a higher percentage of Varroa sensitive hygiene or Minnesota hygienic brood sections were detected and removed by unselected colony, then we would know that brood signaling is also important, correct? And so it turns out that this hypothesis was actually confirmed, and that the brood cues were deemed very important, and they seem to differ from different stocks of bees. So if we summarize up to now what we've seen so far, we've established that colony health is tightly linked to social immunity in honeybees, And that includes both care or prevention and kill or suppression strategies, including high hygienic behavior. We also discussed the different breeding programs that select for hygienic stock and established why these programs can benefit the beekeeping industry by reducing the use of antibiotic and pesticides by beekeeper to treat their colonies against pests and diseases that could be eliminated by using selected stocks. So what does this mean in practice for the beekeeping industry? What you may not know is that many California beekeepers have been selecting for hygienic behavior for over a decade. And Rob Snyder, which is up next, our California field specialist, has been part of the endeavor since the very beginning with BIP. And so I will let him describe how he has been supporting the queen breeders by performing hygienic testing and show you how it's done and what those scores mean. So Rob, you are up.
All right. Just wait for the slide here. So my name is Rob Snyder. I am a honeybee health specialist in Northern California, working with 15 or more queen producers. In 2011, myself and Mike Andre moved to California to assist Katie Lee, who was out here working with the queen breeders about a year and a half before we moved out to assist her and assist the queen breeders by trying to improve their stock through hygienic testing. Prior to moving out to California, Mike and I did a lot of contract work with Jeff Pettis and Dennis Van Ingelsdorf in North Dakota, Florida, Pennsylvania, California, and we were doing colony assessments for health, and we also were doing the National Honeybee Survey, which was a survey that was done throughout the United States. But our overall goal with this project at the beginning and now even is to decrease mortality by improving the queen breeder stock that is shipped across the United States and Canada. So, you know, if we improve the queen breeders stock, the stuff that they ship off to everybody else that uses it, you know, will hopefully decrease the mortality overall in the United States and in Canada, as I said. So here you can see our teams and how they're spread across the United States. The yellow dots are our team's base. And then the orange dots are the beekeeper migratory routes that we follow and assess these colonies for health and look at the pest and pathogen loads and report that to the beekeeper so they can make management decisions. The blue dot is our Maryland lab, which is our main hub for all the samples. All the samples are sent there and they're processed through the same lab, so everything is uniform and done the same way. And you know, our turnaround time from the lab is about 10 days or faster with the samples. And once the samples are processed, they send us the reports, and then we'll analyze the reports and point out anything that is, you know, that doesn't look good, like high mite levels or something like that. And then we'll send those reports off to the beekeepers so they can make management decisions based off the varroa loads and the nosema levels. So we're kind of the boots on the ground as honeybee health specialists, and we're the direct link between scientists and beekeepers. So we get ideas from the beekeepers and kind of relay those back to the scientists so we can work on experiments to try to figure some things out that'll assist them in improving their management strategies. We also do diagnostics and consulting by looking at colonies, inspecting them for health and size. We look at disease and mite loads. We also do research trials where we look at new products, product efficacy. We do emergency visits, which are usually a result of pesticide kills, a bunch of dead bees outside of the front of the hive. And we always ask, you know, if the bees have pollen on their legs or not. If they don't, they're probably getting the pesticide from the nectar. And if they do have pollen on their legs, it could be coming from a plant that was sprayed with the pesticide, and it's in the pollen. Most of all, or most important thing I feel that we do is hygienic testing. And I'll talk about that here in the next few slides. So why do we do hygienic tests? We do this to screen their colonies for the ability to remove diseased or sick brood quickly. Colonies that remove 95% or more are more likely to remove chalk brood, EFB, AFB, or virus-infected larvae, as well as varroa-infected brood. So here's a image of a big crew that really helps when we're doing hygienic testing, and also to have all the colonies in one location. With a crew like this, we can assess 70 to 80 colonies in about 3 hours. We've done about 100 in, like 4.5 hours. And you just take into consideration it's a 2-day thing. So, 70, 80 colonies would take us about 6 hours, because the first day we come out and do the assessment, and then the next day we come back and read the test scores and take samples. So the picture on the left shows a frame being pulled out to assess the colony for brood pattern and the correct age pupa. So what we're looking for when we uncap these pupa on uncap the brood on the frame is a purple eye, which you see in that center picture, or a white-eyed age bee, because we don't want the younger bees where the head's not really developed, because it's more liquidy and it's harder for them to remove, and it's better overall to have like the older purple eye or white-eyed stage. And then on the right image, you can see the PVC pipe. It's 3 inches. It's about 160 cells that are in there approximately. And when we put that into the brood, we push down and twist it. And that kind of smashes the pupa, and it creates, brood juice that will seal that PVC to the wax. And then if there's any open cells, we'll use the squirt bottle, like shown in the right picture, to fill that up before we pour the liquid nitrogen, because that prevents the liquid nitrogen from leaking out and affecting more of the brood, which would make it harder for the bees, because it killed more than that 160 cells. All right, so this left picture shows a frame that we pulled out to be assessed for brood pattern and the correct age pupa. And we depressed the PVC pipe in, and we poured the liquid nitrogen. And on the top right, you can see a video of what it looks like when the liquid nitrogen is boiling off or dissipating. And you could see that those were just poured because the liquid nitrogen is almost at the top of the PVC pipe. We use 8 ounces or more on the broods to make sure that it kills it, because if you don't use at least 8 ounces, it may not kill the brood, and they won't even touch the test. So it takes a lot to be able to kill the brood. So on the bottom right hand image, you can see what it looks like inside the PVC tube after the liquid nitrogen has dissipated. So what we're looking at here, and what we record, is the empty cells and anything that has a crack in it, or a pinhole, or the capping comes off, like you kind of see in the right hand picture at the top. You can see like the rough edges on the cells, and that is due to the liquid nitrogen sometimes will boil the caps off of the brood. So yeah, what we're recording here is just the empties inside of there, and then the next day I'll talk about what we record then. So after selecting the most sealed brood section of the correct age pupa, we depress and twist the 3-inch PVC pipe, as I said on the last slide, into the brood, and those brood juices are what forms that seal. So it's very important to make sure that you have that seal all the way around the edge or otherwise the liquid nitrogen will boil out. If there are any open cells, we use a squirt bottle, like I said, and water just to seal that up. And here you can see after liquid nitrogen dissipates out of the PVC and we count the amount of empties that are there, we'll set all the frames on the side of the truck in the sun so it helps thaw out the PVC pipe, because it can take 15-20 minutes and even longer if it's a colder day. And we're often doing this in the spring before the breeders start grafting. So that's why we set these out. And you can see the brood patterns here on that left picture, how good they are. And that's what we're looking for, is those solid brood patterns. And then on the right picture, sometimes we'll just set the frames out on top of the colonies until they thaw out, and then we'll pull the PVC pipe out, or it'll just fall and drop off if it's sitting on the truck. And then we'll put the frames back into the colony and we will close them up and come back the next day. And we mark the frames with a grease marker or a paint pen, so it's easy to find these frames. And also on our data sheet, we'll mark if it's in the top or the bottom, so we know where we're looking when we come back the next day, so we can kind of stay on schedule with the 24-hour time period in which we want to look at the colonies. So after pouring the liquid nitrogen, we choose the frame with brood at the purple-eyed stage or white-eyed stage. We do a gentle twist with the PVC pipe into the section of the best sealed brood that we can find, which is approximately 160 cells. And then we pour the liquid nitrogen in the tube to do the freeze kill brood. And then we count the empties. And this is just a general overview of what I've already talked about, but I'm just going to run through this just to go through it again. So once we come back the next day after 24 hours, we'll find the test frame, which, like I said, it has the grease or paint pen mark, so it's easy to find. And then we'll count the number of partially removed cells, which are basically anything that's chewed down or there's a perforation in the capping. And then we'll count the number of remaining cells that are sealed. And then what we'll do with those numbers is we put them into our app or into an Excel sheet, and we'll basically be able to generate what the hygienic score is for uncapping, removing, and also partially removing. So we also record additional information on brood pattern, frames of bees, frames of brood, color, which I'll talk about a little bit more. And then also we'll look at the temperament to see, how gentle the bees are. And I've kind of already talked about how we calculate the hygienic scores. So we'll go on to the next frame here, or next slide here. And here you can see on the left a test that was, 4 partials, which you see circled in the green, and then 1 sealed. So that gives us a 97.5, and that's kind of what we're looking for, is something that's very hygienic, 95% or above. And then on the right, you'll see this slide with the partially chewed down stuff and the cappings, and then you'll see a bunch of sealed. So that one's a 63%. So this one probably wouldn't get selected for being a breeder, but this just shows you what each test looks like. And some of them are a lot worse than 63%, which makes it harder for us because it's a lot of counting. So we like to have the 100% ones, because then we don't have to really count. So if they do well in the test, it's easier on the field specialist. So additional information that we record, we look at the brood pattern from 1 to 5. So if you look at these pictures on the right, you'll see a brood pattern that's probably about 1.25 or even a 1. The middle one is about a 3.5. And then the bottom one, of course, is a 5. Very solid, you know, just a few open cells, which are probably bees that emerge. We also look at the bee's temperament, as I said, and the color. So the color is something that's kind of interesting. And people always ask, well, why would you look at the color? And it's from 1 to 5, being 1 being the lightest, 5 being the darkest. But these are kind of the different races of bees. So for the color of a light bee, you're going to see Italian, Minnesota Hygienic, and then Cordovan, which is called like a redhead, but those are probably the lightest. And then you'll move into like the mid grade, which is 3.5 would be like a Saskatraz line, or 5 being dark, or 4, you're looking at Carniolans, Caucasians, and then Russians are probably some of the darkest bees that we see. So we also look at varroa and nosema loads, and we'll look at any signs of disease or pests, and then we give that information to the beekeeper to allow them to be able to make better selection for their breeders. And one interesting thing, people always say, well, how do you test for the temperament? And we don't often do this, but some beekeepers, they'll do a tap test, is what I call it. They basically take their finger and they'll tap a couple times on top of the hive, and then they wave their hand. And if the bees fly out and try to sting their hand, they won't select those as a breeder because that's a sign of aggressive behavior. So here we have a poll. Anne-Marie, if you can help me out.
So we decided to put a poll in to see if you could discern or practice your skills at rating a brood frame. So you will find this this poll, if Nat can put the poll up, asking what brood pattern do you think that is? So do you think that is a 5, a 4, a 3, a 2? The poll is not showing up. So
yeah, I actually don't think I entered that one. I apologize. I think I might have missed that one.
Okay, no problem. So here's what we're gonna do. Everybody in your head, you have to look at this brood and this brood pattern, and remember that 1 is terrible brood pattern and a 5 is the best brood pattern. And so what would you think? And so in your mind, come up with a number.
In the chat box, you can go ahead and select Anne-Marie in the person to chat your answer to, and just really quickly put some numbers in there, and she can quickly scan those numbers. In the chat box, please select Anne-Marie. You have choices. I'm seeing a lot of numbers come into me. It should be going to Anne-Marie.
It doesn't matter because I can't really see it with my slides, but it doesn't matter.
Okay.
We're good, but I'm really appreciative.
We're getting some 4s, some 4.5, some 3s, some 4s, more 4s coming in.
Wow.
It looks like it's ranging between 3 and 4.5s.
All right, with a majority of 4.
I would say yes, a lot of 4s coming in.
All right, so we do this, just so you know, the tech transfer team field specialists, once in a while, we'll send each other, we'll work together and we'll say, hey, what would you call that? Just so we can standardize and make sure that we're all on the same page. We sometimes will send photos through email and just kind of calibrate what we all think. So Rob, what would you say this is?
To me, usually we look at more frames, but I would say this is between 4.25 and 4.5. So probably I would go with 4.25 just because the amount of empty cells that are there.
There seems to be a little bit of something going on here in the middle too. There's a couple of chewed down, it seems like. Anyway.
Yeah, it actually looks like they might have just uncapped those to check the age maybe before hygienic testing. I'm not sure. It could be chewed down brood as well. But yeah, there are some other indications of chewed down stuff that I see there.
All right, good. So this is something that you can do in your colonies to assess your brood pattern and assess the number of frames that you have too. This is kind of a really good colony data. So let's
Yeah,
move on, Rob.
and if you, you know, if the brood pattern is really spotty, it's usually an indication that there's something going on, a disease, or mite levels are high, or you have a failing queen. So you want your brood patterns to look like this and not much worse. I would say, you know, once you're getting below 3, it's there's something going on. In the spring, your brood pattern is not going to be as good because you know, the queen is just starting out. So in the spring, you can be a little more lenient on the queen because, you know, it takes her time to kick into gear and get into full egg laying mode again.
Thank you.
All right. So, you know, after working out here for 9 years, I think I did this 2 years ago. But I had this theory that over time, the hygienic scores were improving and the overall hygienic scores and the consistency of those scores was improving. So I worked with Natalie Steinhauer, our statistician, to look at this information. And you could see on the y-axis the hygienic scores, and then the x-axis is each year, and then each color represents a different beekeeper. So you could see all the scores pretty much going up, except for in 2018, you know, there was a slight decrease in beekeepers 2, 3, 4, 5, 8, and 9. And this was due to a cold snap that happened before, you know, we did the testing. So the queen shut down and stopped laying eggs. And this basically created a break in the brood cycle. So when we came out to test, there was an inadequate amount of nurse bees to be able to clean the test out. And also while we were testing, often the temperatures were below freezing. So in 2018, a lot of queen producers said that it was their worst year in 30 years. So that is what I think contributes to those scores going down in 2018.
Oh, another poll. All right, I guess we want to get back to that question we asked you at the very beginning. How about, are you more likely, or are you likely to purchase hygienic stock of honeybees now that you have gone through this presentation? I will conclude after this.
And another 15 seconds. And just as a reminder, our numbers before were at, well, Anne-Marie, I don't know if you wrote those down. I did. I can tell them at the end if you want me to.
Yeah, that'd be great.
Or the numbers from the previous poll. Okay, we've got almost 80%. Let's try to hit 80% respondents. Let's see if we can get a few more in here.
Yep, 77.
Yeah, we're getting there, getting there.
Yay. Few more. You can do it.
It's good that it's not, I'm seeing not likely. Up, there's one. I jinxed it.
All right.
All right, a couple more votes. Get us to 80. Get us to 80. All right, we did it. Okay,
There we go.
I'm going to share the results, and I'll just talk through what we had before. Earlier, very likely was at 51%, likely was at 24%, neutral was at 20%, and not likely was at 5%.
Okay, great. Looks like we've changed maybe a few people's mind. That's good. I'm glad to hear it. We had maybe some impact. Thank you. All right, so I'm going to let you continue on there, Rob.
So I'm going to try to impact the scores for that last poll a little bit, but you know, since I started working out here, I have not seen sac brood virus at all. And back in Pennsylvania, I would see that, you know, probably 30% of the time when I used to inspect colonies as an apiary inspector. So I think because the bees are so hygienic out here, they're cleaning out that sac brood really quick. And from what I've seen, those hygienic bees, if they encounter a disease, they remove it very quickly and they get that stuff out of the colony. And ones that aren't hygienic, they just kind of, put along, and they don't thrive like a hygienic colony would. And a lot of people think that, hygienic bees are going to not make honey, or they're going to be picking at the brood all the time. But the only time the hygienic bees, pick at the brood is when there's a disease or an issue. So if the colony is healthy, the brood pattern is going to be solid. And some of the most hygienic bees that queen breeders have, that in the operations that we work with, those guys make a bunch of honey. So it doesn't impact the honey production, it seems. And like I said, the only time that that you encounter issues with the hygienic colony is when the brood pattern's spotty. That's how you know something's wrong. So these bees are basically giving you an indicator that you may need to treat with an antibiotic if you have European foul brood or AFB, and that's indicated by a spotty brood pattern. But sorry, I kind of went off on a tangent there, but I'm going to go in the future direction here. The next questions to answer from the database: do increased hygienic score trends over time correlate with disease prevalence of other diseases like AFB, EFB, chalk brood, sac brood virus? And do increased hygienic score trends over the past 9 years correlate with decreased varroa loads and symptoms? So the future direction of Marla's current breeding program is selecting for health and care-kill strategies for disease and mite resistance and local adaptations to floral sources. And other things that will be selected for is winter survival, spring buildup with no feed, hygienic behavior score, propolis and pollen collection, and slow mite population growth.
I can just add a tiny little something, Rob. I took that picture. I was out there in the field with Marla and Dan when we were looking at she was explaining here her prize colony, and her prize queen. She really likes that queen because she's hyperhygienic. She's very hygienic, but also what she's holding in her hand there, she was showing us that she collects a lot, or her bees collect a lot of propolis. So she was really excited that she was starting to see in her bees that were not only hygienic, but also collected a lot of propolis. And so she started to add some of these different criteria for her breeding program. Sorry to interrupt.
So a summary of our presentation here: honeybees are social insects with care-kill strategies called social immunity. 95 are the more hygienic bees detect and uncap, remove disease pupa and larvae at a low stimulus level before infectious stages. And this is important for the industry as it will decrease the use of antibiotics and pesticides used to treat the colonies. And many of the queen breeders that we work with and test and select for hygienic behavior are increasing hygienic behavior scores in the queens they produce each year, which will hopefully impact these queens that are sent all over the country and help other beekeepers have lower losses, and of course try to use less antibiotics or pesticides. So thanks to all our sponsors and collaborators. I'll probably forget some: Project Apis mellifera, California Almond Board, the James M. Fox Foundation, American Bee Journal, Bee Culture, General Mills, USDA, APHIS, Honeybee Health Coalition, Texas A&M, OSU, UC Davis, and University of Maryland. And these are just some of our sponsors and collaborators, and we're grateful for their support in helping us with our research. And
California State Beekeepers Association, CSBA, should also be on there.
Yes, that's correct. But I guess we'll open it up for questions here, and we'll wait and see what you guys have for us.
Great, thank you. Great job. We're going to open up to Q&A. I just want to make a quick announcement that we are going to have another webinar in 2 weeks, so please stay tuned. This one is being recorded, and we will send the link out to our registrants. And so we will send that out, and we're going to open it up to questions. You need to submit your questions by using the drop-down menu in the chat box, the one that says Ask Questions Here. If you've previously sent a question to a presenter, please go ahead and just copy that, paste it into that Ask Questions Here so that Eric can see them all in one spot. And the link is gone, but if you want to support BIP, we do have a donate page on our site to help in supporting what we do and, of course, these webinars. So I'm going to turn it over to Eric, and he's going to start with the Q&A.
Well, hello. Good to see everybody. Thank you for joining us today. Okay, so I do have a couple of questions that have been coming in. First, we're going to start with just some genetic selection questions. The first one came in from Nicole Bega, and she was asking if there is a risk of significantly reducing genetic diversity in these selective breeding programs.
Eric, I'm sorry, could you repeat that?
Yeah, sure thing. Yeah, I was wondering if there, so Nicole Vega asked, is there a risk of significantly reducing genetic diversity in these selective breeding programs?
So, majority of the beekeepers that I work with, you know, to implement their genetic stock, they've used Sue Kobe, who has brought in different Italian genetics from Italy. And she's also, you know, brought in the Caucasian line and the Carniolans. And these queen breeders are implementing this other stock in with their stuff. But a lot of the times they get this stock and it's not up to par with what they have. So they might not use a ton of those different stocks just because their lines are better. But they are trying to implement new stock into their genetic line.
Gotcha.
I hope that answers your question.
And so there's a number of questions asking also, thank you for answering that, Rob. There's a number of questions asking where people can find hygienic queens or how queen breeders typically advertise that, or if they advertise that. We have people in specific areas like Ohio is wondering, you know, if there are any legitimate queen breeders for the VSH line, the VSH line for purchase. So yeah, can you speak to any of those kind of generic, like how do you find out if a breeder is doing hygienic testing? And
So I can rattle off some names, but often these queen producers will say hygienic stock, or Eric, maybe if you could take down those people's names and we could put together a list of the queen breeders that are in the program. We can send that to them. Do you think that would work, or do you want me to just spit off the names that I have now?
I will. I guess probably. I don't know, Anne Marie. What do you think on that one?
Yeah, sure, we can do some of that, but I think that the easiest way would be to find out the origin of, you know, the bees that you're purchasing. And sometimes it's a local vendor, and the local vendor will have to kind of go back into his genetics and explain that. But on the other hand, if you buy a package, you know, depending on where your package is coming from or your nukes are coming from, you can ask the person that is selling you those bees. So, or do you perform hygienic testing? Do you have hygienic scores for your breeders? And just ask the questions about this. A lot of the California queen breeders are doing this. Some of the Georgia beekeepers are doing this, but we do also have other beekeepers that are doing, you know, their own hygienic testing, or having us do hygienic testing for their own bee breeding programs. So there's a lot of that going on. I think that just by asking the questions, when you find out those bees, you know, ask the origin and ask some questions about the stock. I think that's always really important to do, no matter what.
I would say if you're looking for hygienic stock, anything that is produced in Northern California is going to have hygienic traits to some extent, because the hive population density here is very high. So a lot of these guys are sharing drones. And I think that's why some of these queen producers are able to maintain this high hygienic scores in their operations, because these guys are all doing the hygienic testing, and they're all kind of clumped together. And anything from Northern California, in Butte County, Glenn County, Tehama County, Shasta County, any queens that are produced in those areas are queen breeders that have been testing for this for, over 9 years. So I would say anything that's produced in Northern California is probably going to have some pretty good hygienic traits.
If you want to add a little note about drones on that, Rob, because those California queen breeders also have a pretty good, genetically, hygienic behavior bred drone selection as well.
So I can talk on that for a second. So basically, when we do the hygienic testing, they select their breeders, but the ones that just don't quite make the cut, they use those as drone mothers to basically put those out into the yards for the drones. They're like the second best colonies that they're using for the drones, and then they use, of course, the best for the breeders. So they do use those, the ones that don't quite score as well, as drone mothers to improve the drone mating quality.
Wonderful. Thank you so much for answering that, guys. So moving forward, we've got some questions coming in about the pinprick method. Then they kind of ask between if we can compare the freeze method to the pin test method using a needle, and also just if there's any experience among, I guess, Rob or Anne-Marie, if you guys have any experience with those methods.
So you can do the pinprick method, and you don't have to worry about liquid nitrogen, but it does give them a slight advantage because you are putting a hole in the capping. But when you do the pinprick method, you have to make sure you kill the larva. So you almost have to take the needle and stick it all the way through the pupa. And I would do it maybe more than once because they still might be all right. But I haven't done the pinprick method, but if you wanted to do this at your house and didn't have liquid nitrogen or the PVC pipe, you'd be able to tell if your colonies are hygienic or not by comparing, the good and bad. That one slide that I had with the 97.5 score versus the 63. That's basically what you're looking for is ones that clean out the whole thing. So the pinprick method would be fine to do in your own backyard. You just, you just got to make sure that you kill those larvae. But like I said, they are at an advantage to clean those up better because there's that hole in there, and we would count those as empties if we saw like a pinprick in the freeze method. I hope that answers the question.
So just, okay, cool. Wonderful. Yeah, that's perfect. Thank you. So we have a question from Lisa who asked, for the backyard small scale beekeeper who would like to raise queens, is the grid test puncturing the cappings with a pinprick good enough to prove hygienic behavior, or would you say no?
I would say you'd be able to tell which ones are hygienic or which ones aren't fairly easy. And like I said earlier in the presentation, you're looking for 95% and above. So you're going to see, 4 to 7 partials in like one cap or something like that. So if you did that test, you'd clearly be able to distinguish between your colonies, which ones are hygienic and which ones aren't. So I think it's a valid test, and it's easy for a backyard person to do. So yeah, I think you'd be able to find the hygienic ones fairly easy by doing the pinprick test.
Wonderful, thank you. So next, want to move along to a little bit more of like, I guess there's a couple of, actually there's one safety question. Just wondering if there's any safety risk to using liquid nitrogen, and then someone else also asked where could they go to find liquid nitrogen?
So you can get liquid nitrogen at Airgas, but you do need a doer. which is the container to hold it, and they're fairly expensive. They're about $500 to $600 depending on the size, but you can maybe borrow one from somebody if you know anybody in the cattle industry. They often use the liquid nitrogen to freeze the bull semen, so you might be able to borrow one from somebody. But I would say with liquid nitrogen, the risk is, your feet, of course. So you want to wear boots or anything that's not like nylon or anything like that, because if you would pour it, it would soak into your sock and your shoe, and it could, frostbite your foot. You can wear gloves, but honestly, if you somehow poured it into the glove, it would be more of a risk. So if you're pouring it, it just runs right off your hand. It doesn't really freeze you unless you would stick your hand into it. And it does take a little while, to freeze something. So, say you took an orange and stuck it into a cup of liquid nitrogen, it probably takes, 4 or 5 minutes or more for that orange to totally freeze. So it does take some time for things to freeze. So often it just runs off, and the only risk is really your feet. So I recommend wearing a leather boot or something like that. I hope that answers the question.
Yeah, I think that's great. Thank you so much. So a question came in from Karen Pederos. How do honeybees use their venom to protect pathogens from entering the nest?
So they sting. They'll sting. So when we talk about pathogens, of course, they wouldn't do this on a virus or on a bacteria, but they do this on the bigger pathogens. So, they can, so anything that is some sort of a threat to a colony, to either invade for disease or invade for like robbing. So they try to outrun them. They try to use their venom to neutralize them. So they will use venom in order to prevent for those bigger pests to come in. That's how they do.
Thank you. So a question from John Davis asked, what data is available comparing the Harbo specific test for successful mite reproduction versus general freeze kill bees tested the Harbo way? Do you guys know anything of that?
Well, so the important thing to really know, and somebody also asked me a question privately on how to use locally adapted stock. So if you remember what I said, the Minnesota hygienic bees have been bred and developed with the results of the freeze kill assay. But the VSH queen, the VSH program, our bees, have been really developed in bred from low Varroa colonies. So of course, at BIP, we encourage everybody to monitor and to perform Varroa washes of any sort. So, if you want to do the sugar shake method because you don't want to kill your bees, that's better than doing nothing. That's fine. If you want to really, the standard in the industry is doing alcohol washes. I do these washes every month on my colony. And so if you don't want to have to fuss around with the pinprick method or the freeze kill assay or whatever, you could always choose your colonies that are consistently lower in Varroa loads or prevalence to breed from. And so that is one way that others, when we talk about backyard beekeepers, it's not super practical to do the hygienic testing, although it's kind of fun, and it's fun to play with liquid nitrogen, and it's kind of nice to try it. But another way to do this is to achieve it the same way as the Varroa sensitive hygiene has achieved it, by just looking at your Varroa loads and selecting your colonies with the ones that have the lowest Varroa loads. But it takes a lot of generations to do that. So.
Gotcha. And actually, that is a question that came in is about how long would it be if you were selecting this, like if you were on an island, somebody mentioned, how long would it be before you'd start seeing those results?
I think what I would say is, once you start selecting, what I've seen is there's an improvement over 3 years, but it's not a huge improvement, but it's definitely noticeable. You do want to start off with something that's hygienic to try to get, the increased overall hygienic bees out there. I hope that answers it.
Yeah, it does. I don't think that we have like specific data on this and say it would take you so many years, but it also depends on the size of your operation, right? If you do this with a couple of colonies, that's just, you're just not going to be big enough to really make an impact on your breeding program if you want. But I think that, 3 years you start seeing some improvements. But if you look at the stocks for VSH or Minnesota Hygienic, they've been doing this since the 1990s. And this is 2020, right? So we're looking at 25, 30 years in really improving things in a way that is impactful for the industry.
Wonderful, thank you. And I did get a correction there. He was mentioning if you're breeding from the top 90% most hygienic local lines, but I think that the answer would be the same with the data does not exist for that right now. So all right, wonderful, thank you so much. And I think we're going to 1:15. Is that right, Net?
We still have 100 people on the call, so we can go. I think the Zoom is scheduled until 11:30 before it would kick us out. So we can keep this going if there's a lot more questions in your stack.
Okay, there are a couple more questions, but what I'll do is I will go ahead and probably here stop taking new questions because I don't think we'll have time to get into all of them because there are still quite a few that had sent it in earlier.
So Eric, just to comment on something you just said prior to checking on time, and I do think it was presented in the presentation, but I just want to make sure that it's clear. So the California queen breeders in particular will choose breeders or will choose bees that are consistently scoring 95% or more in hygienic behavior. And it's the 95% hygienic that have shown correlation to disease and mite infestation load. So we've got to be careful. It's a correlation, right? So it's not like, we're not saying that if your bees are scoring 97%, that's it, you're resistant to everything, right? That's not what we're saying. What we're saying is that bees that consistently score 95% and above are also consistently the ones that have the lowest amount of diseases and varroa loads. So just a little tidbit.
Cool, thank you. Okay, so now let's see here. We've got, let's see here, back to the next one. So is there any documentation that beekeepers have reduced the amount of treatments on varroa with hygienic bees? That's a question from Fleming.
That's a good question. I believe that Marla Spivak, Dr. Spivak, has a couple of publications that she actually documents and puts science and data behind that claim. Yes, and they've seen it for multiple years, so it's something that has been represented. So yes, it does seem to be published scientific information, and there's evidence for that, yes.
Okay, wonderful, thank you. So next, Sarah Carnicum mentioned the scale you just used is lopsided. So this was at about 1050, so towards the, that's Eastern. I guess maybe that's Pacific, 1030, yeah, anyways. So it was asking, the scale you used, your lopsided, there's 2 positive responses, 1 neutral and 1 negative. You should balance it out for better data by including a very unlikely response as well. And I'm not sure, there is no, I guess, reference to exactly what we were talking about. So hopefully she'll.
No, I know what she's talking about. She's talking about the question that we asked about how likely are you to use hygienic stock. And I have to admit, she's right. I was trying to, not put too many options in the poll, but she's right.
Okay, awesome.
And one more comment about the poll. We did get a comment in that the not likely that was registered was because they don't plan to buy queens, period, in the future. It wasn't because they weren't planning to buy hygienic queens. So we don't really have a not likely selected.
Right. And the thing too is, I guess the thing too here to consider is that this is not just for purchasing queens, but this is for purchasing bees altogether, right? So when you purchase a nuke, you purchase a queen. When you purchase a package, you purchase a queen. So it's not necessarily just going to buy queens. It's also buying any kind of bees and figuring out what kind of genetics you're getting from that.
Gotcha. Yeah, thank you. All right. So we've got another question from Laura Guadalupe Espinosa. She was asking, what is a score of 1 and what is a score of 5 in measuring brood pattern?
So a 1 would be very, very, very poor. Not many sealed cells, not much open brood. You know, your 5 is solid, all sealed, barely any open cells. So, you know, you want your brood patterns to be around a 3 to a 5. If they're below that, I would say there's probably something going on: chalk brood, EFB, AFB, or mite issue, or baleen queen. So, you know, what you want is between a 3 and a 5 for brood pattern, I would say. And those are of course, graded off the amount of sealed brood and open cells.
Wonderful. Thank you. So, James Globa asked, he said, regarding the temperament test, what size hive are they performing a tap test and wave on? 1 through 10 frame, question mark.
The guys that, most of the people I work with, they work with double deeps, so 2 deeps. And yeah, they'll do that tap test. There's only a handful of guys that do it, but I find myself doing it, you know, just to see how they are. But I tap with my middle finger in the middle of the frame, and then I wave my hand pretty quickly above them. And if they don't come at you, you know, they're really gentle, but sometimes you'll do that and you'll get stung a couple times. So be ready to be stung.
That's
tap test.
Do you do this on the frame or on a colony?
On the frame with the lid off. So you're doing it with a open hive. They often do it on the top. They don't usually crack them in half to do it, but sometimes they do it in the middle. But it's just a way to test for temperament.
I can tell you, if I test my colonies, I'll get stung a couple of times if I do that.
Yeah, and sometimes, you know, if they're aggressive, it could be a mite issue. So if they have a lot of mites, they tend to be a little more aggressive.
Yeah, no, it's funny. I've been doing that wrong the whole time. I just kind of go in there and I'm like, oh, they're pretty gentle. I don't antagonize them at all. So good to know.
Yeah, it's just
sometimes they're not.
I mean, awesome. You don't want to kick the hive or something like that. So just a tap a couple times with your middle finger and then wave.
Awesome. Thank you. And an excellent question. Okay, so we already talked about acquiring liquid nitrogen and how long before characteristics. Oh, so Thomas Simpson asked, how long before characteristics decline? First, second generation, etc.
That's a great question.
Yeah.
Wait, what was that, Eric? Sorry.
How many generations are your hives going to have before those hygienic traits start declining?
So basically, like diluting genetics from these breeding programs.
So if we get like a really strong hygienic queen, and then they supersede her, then they supersede her. At what point do you know are we going to start seeing decline in those hygienic traits?
Well, out here, there, a lot of the breeders are breeding for this, so they won't lose it as fast. But as far as like in your backyard, I'm not sure. I have an answer for that one. Maybe Anne-Marie, do you have any idea?
Yeah, it's a tough one. I don't know, because it depends on a lot of factors, right? So it depends on the drones in your area. So if your queen that you receive is 100% hygienic, let's say, and then your supersedure is 50% hygienic, depending on the drones that she's gonna mate with. If they are fairly hygienic, you could have a 75% hygienic queen, right? But if they're not, then you're back down to 50. So it's the game of proportion at some point, and it really depends on your environment and the drones that she's mating with. So it's a difficult one to say, but it could dilute fairly quickly. It could dilute less quickly. It depends. It's not one of those traits that is known to be diluting extremely fast, but at the same time, it really depends on the environment.
Your neighbors.
Yeah, and if you're isolated and you buy all hygienic stock, you're more likely to carry those traits along, especially, like I said, if you're isolated. If you have a lot of other beekeepers, you know, when your queens from your splits go and mate, they're more likely to be diluted if you have neighbors that are keeping bees. So if you do have neighbors that are keeping bees, I would talk to them and try to get them to purchase hygienic stock, and that will increase your chances of keeping that hygienic line going at your house or your home yard.
Gotcha. And we asked somebody, okay, so Ray Goldwire asked, is there a better race of queens that are hygienic, like Russians or Carniolans, etc.?
So that the color doesn't seem to correlate much with the scores. Both of them perform well. I've seen enough to know that they're probably equally as hygienic if the queen producer is selecting for that. So it goes with what the queen producer is selecting for. So a lot of them, I've seen the Caucasians, the Carniolans, and Russians. I've seen them be very hygienic. I've seen the Saskatraz line be very hygienic. I've seen the Italians and the Cordovans and Minnesota hygienic bees. I've seen them with high scores. So the color doesn't necessarily correlate with the scores, and also the colony size doesn't seem to matter. I've seen a 4 or 5 framer clean out the whole test, and it's like a lot of people would think, oh, you need a big colony to remove it, but if they're hygienic, it's going to show. One way you can test it too is throw a paper towel into the hive and come back in a week and see which ones chew it up and remove it quick, because that's a easy way to see if they're hygienic.
Gotcha. Cool. Thanks. That's a fun idea. So, had somebody ask, let's see here. James Gluba asked another question. Have you heard of the work being performed by Arista, an international nonprofit, in developing and refining VSH? They use an evaluation technique of introducing fertile Varroa destructor mites to a colony and pulling pupae to inspect and mite count. I guess that's just a have you heard of it question?
I have not. I've heard about programs where they go and introduce mites and even introduce mites into the cells and kind of look at how the bees, deal with that. But I have not read any, specific recent publication on the subject.
Okay, well, thank you, and James, thanks for bringing that up, because that's something for us to check out. Okay, so a last question here from Laura Guadalupe Espinosa Montaño. How many times do you consider to measure hygienic behavior per colony in order to have a statistically reliable result?
That's a great question.
Eric, I'm sorry. Can you repeat that?
Yeah, sure. How many times do you have to monitor a colony in order to have a statistically reliable result for hygienic behavior?
I would say most of the guys that we work with, we just do it once in the spring, and that's before they select their breeders. So we just do it one time in the spring, and then they go off that. We do test some stuff in the fall, but it's usually artificially inseminated queens that we do that with. Not sure if that answers, or Anne-Marie wants to add anything.
Yeah, well, Rob, no, I want you to add something. So yeah, we test, it's true that we test only once, and we usually test in the spring because it's right before they're gonna choose the breeders that they want to graft from. However, you did mention something about the fact that, like, in 2019, for example, the weather was so crappy that we had much lower scores. So that was not as relevant. So I think that it's important to note that you can maybe test the colony only once, but it would be good to have the best conditions to test that colony. So the best conditions is what, Rob, you can add here is, like, when's the best time to do this test? What's the best time of the season, the best time of the, weather-wise? Can you attest to some of the conditions that make it so that the test is more reliable?
Yeah, so we like to do this in early spring out here. This is going to vary depending on your location, but out here we start in February and continue through March. And basically, this is right when the bees start really laying eggs. So once the days start getting longer, the queens kick in and really start laying eggs. And you look at spring buildup. So you have your breeders selected from the fall, and now you're going through to see which ones are the biggest. And those are the ones that are going to be selected to do the hygienic test on. So I would say early spring in Pennsylvania, you'd probably be looking at March, April, or other places on the East Coast, but it really depends on your climate and your bees building up. So I would do it early spring and look at the biggest ones and do the hygienic tests on those. I hope answers the question.
Yeah, so spring buildup, but does the weather affect it? So for example, if it's cold or if it's nice, if the bees are all flying, is it better that the bees are in? Is it better if there is a honey flow or no honey flow? Any other conditions that matter?
So some of those impact the test, but honestly, if they're hygienic, they're going to be hygienic no matter what. So you're going to be able to see the ones that score the best fairly easy. So nectar flow does kind of impact the test a little bit, but not very much in a negative way. But they are cleaning out space quicker to fill in with nectar. The rain doesn't seem to affect the test, but if it is cold below 0, that seems to affect the test. So if the colony is clustered, they're just worried about keeping the temperature right, and they're not really worried about cleaning stuff up. So if it's below 0, you don't want to really do the test. I would say you want the night temperatures to at least be in the 40s or the 50s.
Excellent. Well, thank you. And that is all the questions we have here. So
Perfect
thank
timing.
you.
We ended right at whatever time zone you're in. Well, thank you to our presenters. I know you're giving them a big round of applause, even though they can't hear you. And please tune in if you want to join us for a webinar in 2 weeks. We'll be announcing that one soon. Stay healthy, and hopefully we'll see you in a couple weeks. Bye-bye.
Thank you.